Well, I'm very pleased to be hosting Dr. Rayane Lima and Dr. Marco Oliveira to discuss their recent PLOS ONE Lab Protocol paper entitled Protocol for the Establishment of a Serine integrase-based platform for functional validation of genetic switch controllers in eukaryotic cells, and the associated detailed protocol in protocols.io. Dr. Rayane Lima is a virologist that currently works with plant genetic modulation tools and metagenome sequencing at the Synthetic Biology Laboratory under supervision of Dr. Oliveira at the National Institute of Science and Technology in Synthetic Biology in Brazil. Dr. Marco Oliveira currently works with genetic modulation tools in eukaryotic and prokaryotic systems, including Mycoplasma synthetic minimal genome and metagenome sequencing at the Synthetic Biology Laboratory, also under supervision of Dr. Oliveira. Marco, Rayane, thank you so much for being with me.
Thank you for having us.
So to begin with, can any of you explain briefly what the protocol is about, how to use it, and why it is relevant?
Yes, this protocol was designed to enhance the serine integrase-based genetic switches in eukaryotic cells. It outlines a platform for creating and screening the switches, which control gene expression through directional inversions of DNA sequence orientation. The system involves two sets of plasmids, one effector module and a reporter module, both incorporating regulatory components suitable for expression in mammal cells, human cells, and plant cells. The complete methodology includes plasmid design, DNA delivery, and subsequent molecular and phenotypic assessments. And this protocol is relevant because it provides a structured workflow for identifying and validating new integrase-based genetic tools.
And to understand the significance of the protocol better, can you describe what was the motivation to develop the protocol? So were there existing similar protocols? And if so, why they didn't work for you?
The primary motivation to optimize the protocol was to increase efficiency and reduce the time required for each step, ultimately saving resources and allowing for more rapid results. Lastly, as our project grew, it became essential to optimize the protocol to handle larger data sets or more complex cell samples efficiently.
Yeah, and it's not like the previous ones didn't work. It's more that we had to work with a variety of eukaryotic systems. We have animal cells, human cells, plant cells. So we had to develop a system using different integrates with different efficiency activities. So therefore, to consolidate all the methodology for each model, we had to prepare a very robust protocol in which we outline every and each critical steps for each one of the models. So it's like the other ones work, but we have to compile everything together and adapt it so it could be more, we have more plasticity for different models and different effectors, integrases.
And when you were developing and optimizing the protocol, what are the most critical steps for a successful performance?
So in our systems, one of the most critical steps was the design and assembly of the plasmids, including the selection of the regulation modules like promoters, terminators. We, for instance, decided not to work with some of the candidate integrases because the recombination sites could add initiation code, like start codons upstream of the gene or the real starting codon, like the designed start codon. So that could potentially disrupt gene expression. Therefore, also attention to details are very crucial in this step because it sets all the platform. And another crucial step is the transfection process for delivery of the system, both integrase and the reporter system. The system requires maintaining healthy, high quality cells and performing the procedure with very patiently and with high precision. So transfection, that can be a laborious and tiring process, but it's essential for the successful genome editing. Ensuring optimal conditions and careful managing each step helps to maximize the efficiency and accuracy of your results, your project.
And when you were developing the protocol, was there anything that you tried and didn't work and didn't make it to the final version of the protocol?
Yes, one approach we explored was adding NLS, nuclear localization signals, to the integrases in an effort to enhance their performance. The idea was that directing the integrases more effectively to the nucleus, it could improve their efficiency. However, after thorough testing, we found that the results were inconclusive. The performance enhancement we hoped for was not consistently observed. And as a result, we decided not to include this modification in the final version of the protocol. Integrases, in their native form, are inherently capable of assessing the nucleus, where they perform the genome editing. So our research demonstrated that the absence of NLS did not significantly impact the main findings of our study. So this finding underscores the robustness of the integrase's natural ability to engage, to target the genome, making the inclusion of an NLS an unnecessary modification for our protocol. So we completely removed these studies.
And both your protocol and the paper are relatively young. But still, I wonder whether since the time of publication, have you made any further optimizations or modifications of the protocol, or have you tried applying it to a different organism or cell type?
Yes, definitely. We developed a plant genome integrated bimodular system driven by 4 integrases. We denoted the system as INTPLEX. This system allows us to perform DNA editing through both inversion and excision, both DNA inversion and DNA excision. These enhancements have made the system more versatile and efficient, enable more target and reliable edits in the plant genome. The improvements were driven by our ongoing goal to refine the system for better performance and broader application in plant biotechnology. We are submitting this manuscript now.
Yeah, I think like the main difference compared to the protocols we've published is that now instead of transient expression, we have the system integrated in the genome. So
in the plant genome.
Well, and it's very exciting that you are making a paper out of it as well.
Yeah.
Congratulations.
Thank you.
Thank you.
And good luck with the peer review process as well.
Yes.
And now I actually want to talk about that. So you are doing it again, but what was the motivation for you to send the first protocol to become a peer-reviewed paper?
So the motivation to publish the protocol, especially in a protocol, in a platform specifically for protocols standardized, like we have the lab protocols and the protocols IO, it came from our strong belief that the potential use of serine integrase as DNA modulators, even like in other systems that we already work with, we saw a clear need to share that with the scientific community and make the protocol itself, or the whole platform, more consolidated and standardized to facilitate the application of these methods and these integrases in very different models depending on which the other groups are working with, or also testing new integrases that we expect that all the time we have more new integrates being described and found in nature. So we have a platform that they can be easily tested and their activity assessed in eukaryotic systems, since they originally come from bacteria and prokaryotic organisms. We need a interface to change the cell model. And the peer review process, they also provided an opportunity to validate and refine our work and ensure the reliability and usefulness of the process in other fields.
And what was or how the feedback that you received from the peer reviewers helped you improve the manuscript and shape the final version of both the paper and the protocol?
Well, one of the key contributions was in improving the clarity of our graphical analysis. The reviewers pointed out areas where the data presentation could be more intuitive and accessible, which led us to refine our diagrams, some of our diagrams. This not only made the protocol easier to follow, but also enhanced the overall communication of our results. Their insights helped us to present our work more effectively, ensuring that other researchers can better understand and apply the protocol in their own studies.
Yeah, and the nature, like the dual nature in the system in which we have the protocol step by step published in the platform, and we have the manuscript published as a paper on the lab protocol PLOS ONE, is that it allows you to not only give the instructions, but also in the manuscript you can do all the consideration and theoretical aspects of it and how you can adapt and adjust, not only just giving the step-by-step instructions. It enriches the process very much.
It allows you to contextualize
Exactly.
the protocol and to add discussions and limitations if there are any as well, right?
And that's something that the reviewers helped a lot of, like how to pass this information in a more contextualized, as you said, manner.
And again, this is a young protocol and a young paper. So the question might be still speculative, but how do you think the protocol and the paper will impact your careers as young scientists?
This protocol has been incredibly significant for our career, as it marks the first time we've developed such a robust methodological protocol. So the process of creating and refining it has deepened our understanding of the intricacies involved in scientific research and protocol development. And it's been it's been a pivotal experience, allowing us to contribute meaningfully to our field and providing a strong foundation for future research projects. The skills and knowledge we've gained through this work have already begun to shape the direction of our career in a very positive way.
Yes, talking about skills and knowledge, I think the important part is that since it's such a broad platform with like many different organisms, we had to work with many collaborators and like different groups from other states in Brazil. With special like some are specialized in stem cells, the other ones at cancer cells and patient cells. So doing like the management of the whole group was also a very interesting opportunity to develop like this ability, not only to do experiments and apply our knowledge, like a formal knowledge we already have, but learning how to adjust a bigger project in a multi-component team. It's like very positive.
Well, that was my last question. So I want to know if there's anything else that you would like to add to this conversation that we haven't discussed so far.
I think we covered many bases. One thing that I believe I would like to say it's how I was working with the team for the submission, the team like the platform, the protocol entry service, because since we had like so many, it's not only a single protocol, it's a group of protocols and a group of protocols for different organisms. We were not knowing exactly how to publish that, how to structure that. And they helped us having like the opportunity to create not only a single entry for many different organisms, but different entries and organize that as a protocol collection in the page. It was like very helpful in knowing how to make public all our methods. That was like very good. Thank you.
And many thanks for the opportunity. I think we talk about the
Yeah, and so.
Well, Marco, Rayane, thank you so much for carving some time in your busy schedule to talk to me. It was very enlightening, the conversation. I think it was very clear, and I appreciate very much your time.
Did you? Thank you. I'd like to thank God the opportunity, not only but they had the opportunity to make the protocol public, but now it's just talk about it. I think it's a very, very important thing. Thank you.
It was a positive experience, man. Thank you for this, for having us today.